Hello, I'm Montana Smith, Metadata Coordinator for the Environmental Molecular Sciences Laboratory. And in this video, I am going to talk about the value of metadata, including what it is, why it's important, and DOE and EMSL's data policies. When we say metadata, we mean contextual data about your data. For this video, we will be describing sample metadata. This covers the when, where, what, and how of the samples you've collected during your experiment. So why collect metadata? Well, it has many benefits to your immediate research, long-term data preservation, and scientific discovery. For your immediate research, it ensures that your samples and data are captured correctly and without confusion. It also helps EMSL scientists understand your samples and project goals so samples are properly handled and data is correctly analyzed. Remember, our scientists are thinking about a lot of projects, and we want to be sure that yours is clear and understood. Clear and concise metadata will improve data and sample interoperability for long-term projects, cross-study questions, and analysis across multiple data types. All this also ensures that data can be reused, which allows for bigger science and extended research questions. Good data management ensures the effective reuse of data and integration of knowledge by the community after release. Science goes through a data lifecycle of question, creation, processing, analysis, preservation, access, and finally, reuse. With reuse leading to expanded and more informed research questions or additional data curation, which expands knowledge, discovery, and innovation. Overall, the goal of good data management, metadata capture, and standardization is to enable great science. To accomplish this, in research, it means being intentional about collecting, storing, processing, and protecting your data. For data preservation, it means making data fair, findable, accessible, interoperable, and reusable. Unfair data are impossible to understand or track, and without fair, data sharing and interoperability wouldn't be possible. Which is what we're faced with today. Many fields of research have a data cycle that look like this: spaghetti science that is hard to map, often across even our own experiments, let alone across different experiments, labs, or research fields. So the goal is to get to here, with clear connections between source and target, clear data mapping with easily relatable findings that allows for direct data comparison. And we can do this with standardized metadata. Now, there's a lot of types of metadata out there, and many things to think about when we say standard metadata. Step 1, and in the context of this video, we are focusing on sample metadata. This is the experimentally unique sample. Managing metadata by category and class provides streamlined relationships and builds upon itself. There are 4 classes we're separating metadata into. Sample metadata describes and provides all the information one would need to understand the individual starting material, the sample or biosample, the environmental or synthesized materials that are generated in your experiment. A lot of standardized metadata fields and ontologies used in the EMSL sample metadata collection have been developed by the Genomic Standards Consortium and in collaboration with the National Microbiome Data Collaborative, making data and metadata interoperable. When thinking about what metadata is important for your project and your samples, you can ask yourself, what would someone need to know about the sample to understand it, and how do the samples differ from each other? Metadata should describe each sample, how each sample differs and is unique by capturing treatments and distinguishing conditions. This can include where the sample originated from or how it was generated, also identifying replicates where they exist. Metadata should also provide information about what analyses are planned for the samples and if there's any special considerations to be made, such as substrates or isotope labeling or storage conditions or processing. Metadata collection and standardization is done in accordance with our sponsors' policies. The Department of Energy, Office of Science requires that data generated be made publicly available. This isn't a new requirement, and EMSL has always had a data release policy. EMSL balances its users' needs with sponsor expectations to make data openly accessible to the public as soon as is reasonable. This includes making users' data accessible, immediate, and persistent. Data and the corresponding metadata generated at EMSL becomes publicly available when any one of the 4 events occur: at the time of publication, when a data DOI is minted, at the discretion of a project team member at any time, or 1 year after data generation. Any publications produced using the openly accessible project data are expected to cite the EMSL project DOI and the data DOI, so credit and recognition are given to the PI and team members. Data as it relates to the data management policies here includes sample metadata, raw instrument data, associated experiment metadata, and process data. Again, released at a minimum of 1 year after data generation. In summary, our goal at EMSL is to provide clear and interoperable metadata and data sharing to elevate science and expand knowledge and discovery. I hope you found this video helpful to demystify metadata and EMSL data policies. For additional trainings, you can find more videos at the EMSL at PNNL YouTube channel or at emsl.pnnl.gov. You can also contact your project manager or metadata point of contact for more information or questions. Thank you.